Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
If you contract an Orthoebolavirus, you are essentially flipping a coin on your own life.
Right. It is literally a coin toss in terms of survival.
Exactly. I mean, we are talking about an average case fatality rate of roughly 50%.
Mm-hmm.
It is easily one of the most terrifying pathogens on the planet.
Mm-hmm.
And yet, our primary licensed medical defenses vaccines, like Ervebo and Zebdino, essentially only work against one specific strain.
Which is the Zaire Ebola virus, and that is a staggering vulnerability in our global health infrastructure.
It really is.
We have this incredibly lethal viral family, and our absolute best shield really only covers a fraction of the overall threat. If a different strain spills over tomorrow, those stockpiled vaccines are virtually useless.
But what if that is about to change? I mean, what if the exact same mRNA technology that completely revolutionized our response to COVID-19 could be engineered into a broad spectrum, all-in-one defense shield?
A single regimen that covers the whole family.
Right. We are talking about training the immune system to hunt down 3 distinct, highly lethal Ebola strains simultaneously with robust protection, lasting well over a year from just a single shot.
To really grasp the real-world stakes here and why the scientific community is so intensely focused on this, we have to look back at recent history.
We need to set the context for why this matters right now.
Exactly. Think about the 2013 to 2016 West African outbreak. That was driven by the Zaire strain, and it was absolutely devastating.
Over 11,000 deaths, if I remember the numbers correctly.
Right, over 11,000. Entire communities were just decimated, and healthcare systems were pushed way past the breaking point. The sheer terror of that specific outbreak actually accelerated the development of the vaccines we rely on today.
It was a massive triumph of science to get those vaccines across the finish line so quickly.
It was. We built the weapon, and it worked against Zaire. But then, we fast forward to just a couple of years ago, the 2022 to 2023 Sudan virus outbreak in Uganda.
And suddenly that triumph felt a lot more fragile.
Because it was. That outbreak in Uganda exposed a massive blind spot in our preparedness. The Sudan strain of the virus is structurally different enough from the Zaire strain that our current vaccines offered little to no cross-protection.
So public health officials were essentially fighting a new war with weapons designed for an old one.
Exactly. They had to basically start from scratch, scrambling to get experimental vaccines into clinical trials while the outbreak was actively spreading through the population. It highlighted a terrifying reality, which is that chasing these viruses one strain at a time is a losing game.
And that is exactly what we're exploring today. We are unpacking a groundbreaking new paper from the Proceedings of the National Academy of Sciences, or PNAS.
A highly respected journal, so the data here is incredibly rigorous.
Very rigorous. The researchers outlined a multivalent mRNA vaccine called GPs+NP at LNP. And whether you are a healthcare professional tracking epidemiology, or a science enthusiast fascinated by mRNA, or just someone looking to understand how we actually prevent the next pandemic, this deep dive is going to reveal exactly how scientists are rethinking vaccine design from the absolute ground up.
The paradigm shift we are talking about here is moving from reactive vaccinology to proactive vaccinology.
Let's define that for the listener. Reactive meaning waiting for the fire to start.
Right. Reactive is where we wait for an outbreak to start, identify the strain, and scramble to build a bespoke vaccine. Proactive means we want a vaccine already on the shelf that anticipates the broader threat.
But to attack multiple strains simultaneously, you cannot just throw a bunch of random viral pieces at the immune system and hope for the best.
No, not at all. You have to really understand how the virus operates on a fundamental structural level.
Okay, so let's get into that anatomy. When we talk about Ebola or any of these Orthoebolaviruses, what is the immune system actually seeing when the virus enters the body?
Well, the virus relies heavily on glycoproteins, which we often just abbreviate as GPs.
The outer spikes.
Yes. If you look at an electron microscope image of the virus, these are the outer spikes protruding from the viral envelope. They are the chemical keys the virus uses to latch onto and infect our human cells.
So naturally, these glycoproteins are the primary target for our vaccines.
Exactly. The immune system sees that spike, recognizes it as a foreign invader, creates an antibody specifically tailored to that exact shape, and neutralizes the threat before it can even enter our cells.
That sounds straightforward enough on paper, but I'm guessing it is not quite that simple in practice.
Far from it. The primary problem is that these glycoproteins mutate. They vary wildly between the different strains. So Zaire, Sudan, Bundibugyo, they all have different spikes.
So the spike on the Zaire virus looks completely different to the immune system than the spike on the Sudan virus. Wait, so if I am understanding this, let's think about this like a getaway car used in a bank robbery. The glycoprotein, that outer spike, is basically the paint job and the license plate.
I like where you were going with this analogy.
So the Zaire strain might be a red car with a very specific license plate, and then the Sudan strain is a blue car with a completely different plate. And the Bundibugyo strain is a black car.
Right. They all look different on the outside.
So if you train the immune system, let's call them the police, to only look for the red car with that one specific Zy plate, the blue and black cars are going to drive right past the checkpoints.
They will completely evade detection. That analogy perfectly captures the core biological challenge here. If you only target the paint job, the virus evades you simply by changing colors over evolutionary time.
So how do you catch all the cars?
You look under the hood. Underneath the hood of that getaway car is something called the nucleoprotein, or the NP.
The engine block.
Exactly. The nucleoprotein is the engine block. It is an internal structural protein. And unlike those outer spikes, it is highly conserved.
Meaning its genetic sequence doesn't really change much.
Right. It is nearly identical across all these different Orthoebolavirus strains.
Yeah.
It plays a highly crucial role in forming the nucleocapsid, which is the structure that protects the viral genome itself.
So the virus absolutely needs this specific engine to replicate.
It does. It cannot just mutate away from you without destroying its own ability to function as a virus.
That makes total sense. So if the engine block is identical across all the strains, why haven't we just been making vaccines out of the nucleoprotein this whole time? Why even bother with the mutating glycoproteins at all?
That is a great question. The issue is that the immune system doesn't easily see the engine block when the virus is just cruising down the bloodstream.
Because it's hidden inside the car.
Exactly. The nucleoprotein is tucked away deep inside the viral envelope. The antibodies circulating in your blood are looking for surface features. They're strictly looking for the paint job.
So if you only vaccinate with the nucleoprotein, you don't generate those crucial neutralizing antibodies.
Right. You miss the opportunity to block the virus from entering the cell in the first place.
Ah, I see. You need to stop it before it gets inside.
Mm-hmm.
But if it does get inside, you want the immune system to immediately know what the engine looks like so it can destroy the whole factory.
Precisely. And the researchers behind this PNAS paper recognized that targeting just the glycoproteins was insufficient for broad-spectrum protection, but targeting just the nucleoprotein wouldn't stop the initial infection.
So they decided to do both.
Yes. They engineered a highly specific 4-part mRNA payload.
Let's break down that recipe.
They created messenger RNAs, mRNAs, encoding 3 completely different glycoproteins. So one from Zaire, one from Bundibugyo, and one from Sudan.
That is 3 different paint jobs, so the immune system recognizes all the cars.
Exactly. And then they added a 4th mRNA, encoding that highly conserved nucleoprotein from the Zaire strain.
The universal engine block, so it is 4 distinct blueprints. But they didn't just pull these blueprints off a shelf, did they? Because when I was looking through the methodology section of the paper, the amount of engineering they did before they even put this into an animal model was just wild.
It really highlights the precision of modern vaccinology.
They didn't just grab the first Zy glycoprotein sequence they found on a database?
No, they were incredibly meticulous. And honestly, this is the beauty of the mRNA platform. You can tweak the genetic code at the molecular level to optimize exactly how the human body reads it.
So they went through a rigorous screening process just for the Zaire glycoprotein.
Yes, they actually synthesized and tested 4 completely different versions of the Zaire blueprint just to see which one the human cells would build the most efficiently.
What kind of versions are we talking about here?
Well, they started with a wild-type version.
Which is just the exact genetic sequence as it appears in nature, right?
Exactly, the raw code. Then they tried a second version interspersed with a specific signal peptide, which is designed to help route the protein properly inside the cell.
Okay, a routing tag.
Then they tried a third version that was engineered to form stabilized trimers, essentially trying to force the protein to hold its optimal 3-dimensional shape so the immune system gets a good look at it.
And the 4th version?
The 4th version was called MOLDEL.
MOLDEL. Okay, what exactly does that stand for?
It stands for a full-length glycoprotein lacking the mucin-like domain.
Mucin-Like domain. You're going to have to break that down for me because it sounds like something involving mucus.
You are actually not far off at all. The mucin-like domain is a highly glycosylated region on the viral spike.
Glycosylated meaning covered in sugars.
Yes. Glycosylation is a biochemical process where sugar molecules are physically attached to a protein. So this mucin-like domain is essentially a dense, messy shield of sugar molecules that the virus wears on its surface.
Why would a highly lethal virus wear a shield of sugar?
To hide. Our own human cells are covered in various sugars. It's a normal part of our biology.
So it's camouflage.
Precisely. By draping its most vulnerable, recognizable structural features in a thick coating of sugar, the virus camouflages itself. The immune system has a much harder time recognizing the underlying viral protein because it is obscured by this thick, sugary fog.
That is deviously clever on an evolutionary scale.
Yeah.
So the researchers thought, what if we just delete the genetic instructions for that sugar shield?
Exactly. By deleting this mucin-like domain in the mRNA construct, creating that MOLDEL version, they stripped away the camouflage completely.
But they had to be careful with that, right?
Very careful,
Yeah.
because sometimes when you delete a massive chunk of a viral protein like that, the whole thing folds incorrectly.
And if it folds wrong, the immune system learns the wrong shape, and the vaccine is useless.
Right, so they had to test it rigorously.
How did they know it actually worked?
They transfected these different mRNA constructs into human cell cultures in the lab. They essentially hijacked the cell's machinery to see how well they would read the blueprints and manufacture the protein.
And then the results.
When they measured the expression levels, the MOLDEL version was the clear winner. It showed significantly higher expression levels, meaning the cells produced much more of the protein without sacrificing any specific antibody titers.
So the antibodies produced against the naked MOLDEL version still perfectly neutralized the real, fully shielded virus.
Yes, they did. It was a complete success.
That is just a brilliant piece of molecular engineering. They optimized the antigen blueprint before they even thought about how to deliver it to the body. They made the instructions easier for our cells to read and the resulting protein easier for our immune system to target.
It is elegant science.
So the final recipe was this highly optimized MOLDEL-Zaire glycoprotein alongside the Sudan glycoprotein, the Bundibugyo glycoprotein, and that conserved nucleoprotein engine block. 4 distinct optimized instructions.
Right. But having the perfect blueprints is really only half the battle. This brings us to a major hurdle in mRNA technology. We know what is being delivered, but how you package those mRNAs dictates the entire immune reaction.
Right, because if you just inject raw mRNA into the bloodstream, it doesn't just casually float around and find a cell, right?
No, it will be destroyed almost instantly.
Why is that?
Our blood is absolutely full of enzymes called RNases. Their entire job is to seek out and shred free-floating RNA.
It's an innate defense mechanism.
Exactly. From an evolutionary perspective, free-floating RNA usually means a virus has burst open nearby, so our bodies are trained to clear it out immediately. To get the mRNA safely into our cells without being shredded, it requires a protective shell.
And we call this a lipid nanoparticle, or an LNP.
Right. It's basically a microscopic fat bubble that acts as a stealth delivery vehicle.
Okay, but the researchers faced a really critical design choice here. And this is where the paper gets extremely fascinating from an analytical and immunological standpoint.
It is the crux of the entire study, really.
I was actually wondering about this when I read it. If you have 4 different mRNAs, why go through the massive headache of cramming all 4 of them into a single lipid nanoparticle?
It is biochemically very difficult.
Right. Why not just manufacture 4 separate vaccines, 1 for each mRNA, put them in their own separate little fat bubbles, and then just mix them together in a vial right before you inject the patient? It seems like it would be infinitely easier to manufacture and quality control.
You are absolutely right from a manufacturing standpoint. Making one LNP with one mRNA is a very well-established, straightforward process right now.
We do it for COVID vaccines all the time.
Exactly. But trying to balance 4 different mRNAs inside a single lipid nanoparticle without them chemically interfering with each other is incredibly complex. Separate encapsulation seems like the obvious, easy choice.
But immunology does not always favor manufacturing simplicity.
It rarely does, and the body reacts drastically differently depending on how those packages are bundled.
And the researchers didn't just guess at this. They explicitly set up an experiment to test this exact question.
Yes, they created 2 distinct formulations to compare. The first was the all-in-one approach. That is the 3 glycoproteins and the nucleoprotein all co-encapsulated inside the exact same lipid nanoparticle.
All 4 blueprints in one bubble.
Right. And the second formulation was the mixed approach. Each mRNA packaged into its own separate LNP, and then those 4 distinct nanoparticles were physically mixed together in the syringe.
Okay, let's walk through what actually happens when that mixed syringe hits the human immune system.
It becomes a game of chance.
Right. If I have 4 different bubbles floating around and they start bumping into my cells, it's just basic probability. One cell might absorb the bubble with the desired blueprint. A completely different cell, maybe further down the arm, might absorb the Sudan blueprint.
Precisely. And you end up with what we call an erratic or asynchronous immune response.
Because the cells aren't coordinating.
Right. Because the mRNAs are packaged separately, there is absolutely no guarantee that a single antigen-presenting cell in your immune system is going to process all 4 viral proteins at the same time. You get these massive, unbalanced, antigen-specific variations.
And why is that biologically bad? I mean, don't you eventually get antibodies against all of them anyway?
You do get antibodies, but the quality and the balance of that immune response really suffer.
It's uncoordinated.
Exactly. Furthermore, the researchers noticed something really troubling when they analyzed the immune data from that mixed nanoparticle approach. It leaned heavily toward a strong but incredibly narrow cellular response.
It was skewing the whole system.
Yes. It was skewing certain cytokine secretions, which are the chemical messengers of the immune system, in a way that just wasn't optimal for long-term, balanced, broad-spectrum protection.
But when they used the all-in-one approach, the single bubble.
The all-in-one lipid nanoparticle forces what we call synchronous delivery.
Synchronous delivery.
Yes, because all 4 mRNA blueprints are literally trapped inside the exact same microscopic fat bubble. When a host cell engulfs that LNP, it is forced to take in all 4 instructions simultaneously.
There is no probability involved.
None. That single cell is forced to translate all 4 proteins and present them on its surface at the exact same time.
And the immune system prefers that synchronous presentation.
It completely thrives on it. Synchronous delivery is the absolute key to immunological synergy in this vaccine. Inside the single lipid nanoparticle group, the researchers observed a beautifully balanced Th1 and Th2 polarization.
Okay, Th1 and Ph2. I want to make sure we ground this for the listener. Let's dive into Michael's science minute here to break down this dual-pronged immune response.
Let's do it.
We are talking about 2 different types of helper T cells that basically act as the generals of the immune response, directing the troops.
That is a highly accurate way to visualize it.
So Th1 cells generally drive the cellular response. They are the ones that activate the assassins, the CD8-positive cells that hunt down and kill infected host cells.
Right. They handle the internal threats.
And Th2 cells, on the other hand, drive the humoral response. They act as the architects, instructing the B cells to build massive amounts of neutralizing antibodies.
Exactly. So if Th2 cells are the architects building the antibody wall, and Th1 cells are the frontline commanders hunting down the infected cells, the all-in-one nanoparticle perfectly balanced these 2 forces. Neither one overpowered the other.
It created a perfectly symmetrical defense strategy.
It did. And beyond just balancing those forces, synchronous delivery achieved something even more critical. It forced the B cells to recognize the nucleoprotein.
The engine block.
Yes. The nucleoprotein is traditionally considered a recessive antigen.
Recessive meaning it gets ignored.
Mostly, yes. When the immune system encounters a virus naturally, the B cells are highly distracted by the flashy, protruding surface glycoproteins. They are so incredibly busy reacting to the paint job that they often completely overlook the internal nucleoprotein.
It's like the police being so focused on the bright red car that they don't even bother to look under the hood to see how the engine works.
Exactly right. But by forcing the host cell to manufacture and display the glycoproteins right alongside the nucleoprotein, literally coming from the exact same delivery vehicle at the exact same time the immune system was essentially cornered.
It couldn't ignore the engine block anymore.
Right. It was forced to process it. The B cell recognition of the nucleoprotein was significantly enhanced compared to when the mRNAs were delivered in those separate mixed nanoparticles.
This perfectly sets up the dual-pronged immune response that I really want to dig deeper into. We've talked about the humoral side, the antibodies, and the cellular side, the T cells.
Yeah.
Let's spend a minute diving deeply into why combining the glycoprotein and the nucleoprotein is such a massive game changer for surviving an actual lethal infection.
This is where we really see the true elegance of the adaptive immune system working in concert. Let's start with the humoral arm.
The antibody wall.
Right. The mRNA blueprints for the 3 glycoproteins trigger the B cells to create IgG antibodies.
And these antibodies act like a physical barricade in the blood.
They do. They circulate in the blood, bind to the outer spikes of the Orthoebolaviruses and block them from ever docking with and entering our host cells.
They basically jam the key so it can't go into the lock.
Exactly. That is the ideal scenario. Sterilizing immunity. But here is a truly fascinating twist buried deep in the data of the paper. The researchers noted that the IgG antibodies specific to the nucleoprotein were actually somewhat inhibited when they were co-expressed with all those dominant glycoproteins.
Wait, inhibited. So the vaccine produced fewer antibodies against the engine block than they initially expected.
Yes. The glycoprotein response was so dominant, so loud to the immune system, that it actually suppressed the antibody response to the nucleoprotein.
Now, on the surface, that sounds like a massive failure of the design.
Hmm.
I mean, if your whole goal was to force the immune system to recognize the nucleoprotein and the antibodies underperformed, didn't the all-in-one design fail?
It definitely sounds like it until you connect it to the bigger biological picture.
Okay, let's connect it.
It didn't matter at all that the nucleoprotein antibody response was low, because the nucleoprotein is an intracellular antigen. It lives strictly inside the virus and eventually inside the infected cell.
And antibodies can't easily get inside a cell to do their job anyway. They're too big, right?
Right. The nucleoprotein's primary role in this specific vaccine design is not to build the antibody wall. Its role is to activate the CD8-positive killer T cells.
Ah, the cellular arm. So the antibodies are the wall, but the nucleoprotein is there strictly to train the assassins.
Exactly. The nucleoprotein trains the killer T cells to hunt down and destroy any host cells that have already been breached by the virus. And the data from the study on this specific cellular mechanism is just incredible.
Let's talk about that data because the methodology here is fascinating. How do you even measure the activity of killer T cells in a lab setting?
They use something called an ELISpot assay.
Break that down for us.
They took the spleens from the vaccinated mice. Now, the spleen is a major hub for immune cells. It's like a central command center. And they isolated the splenocytes, the immune cells, from the spleen.
Okay.
Then in a petri dish, they stimulated those immune cells with highly specific pieces of the nucleoprotein to see exactly how they would react. They were literally counting the exact number of T cells that started secreting specific cytokines like interferon gamma and interleukin-2.
Cytokines are basically the chemical flares that T cells shoot off when they recognize a target and initiate an attack. Right?
Exactly. It's how they communicate that a threat is present. And the petri dish just lit up. Nuclear protein stimulation resulted in over 200 of these cytokine spots per 500,000 splenocytes.
Is 200 a lot in this context?
To put that in perspective, when they ran the exact same test but stimulated the cells with the glycoproteins, it reached a maximum of only about 150 spots.
Oh, wow. So the engine block was driving a much stronger response.
Yes. The engine block, the nucleoprotein, was driving a vastly superior cellular attack. It was pulling far more weight in recruiting those killer T cells than the spike proteins were.
Okay, so let me summarize this mechanism to make sure I have it locked in for the listener. The glycoproteins create this massive wall of circulating antibodies. They're the first line of defense. If the virus enters the bloodstream, the antibodies smother it before it can dock with a cell.
Correct. That's the humoral shield.
But viruses are incredibly efficient, and Ebola in particular is notoriously aggressive. So if even a few viral particles manage to slip past that antibody barricade and infect a cell, the glycoproteins cannot help anymore. The antibodies are essentially useless once the virus is inside the cell.
Exactly right. They can't cross the cell membrane.
That is when the nucleoprotein-trained killer T cells take over. They act as a highly specialized internal security force. They constantly scan the body, identify the chemical signatures of the compromised cells, and trigger them to self-destruct before the virus can replicate and burst out to infect 100 more cells.
It is a true, robust, multi-layered defense. You are blocking the doors with antibodies, and you have an elite security team sweeping the rooms with T cells.
So we understand the microscopic cellular level perfectly now. We know exactly how the antibodies block and how the T cells destroy. But a vaccine can look absolutely incredible in a petri dish or in a blood draw and still completely fail to keep a living organism alive against a lethal viral exposure.
The true test is in vivo.
Right. We need to look at the macroscopic survival outcomes.
And this is where the PNAS paper moves from elegant molecular engineering to absolutely undeniable efficacy. The researchers started with a mouse-adapted Ebola virus model. Specifically, they used the lethal Zaire strain.
Mouse-adapted just means they tweaked the human virus slightly so that it actually infects and kills mice, right? Since human Ebola doesn't naturally infect mice.
Exactly. It allows them to study severe disease pathology in a small animal model. So they took mice that had received this multivalent mRNA vaccine and exposed them to a completely lethal dose of this virus.
A dose guaranteed to kill a normal mouse.
Without question.
And the survival results.
Absolute, complete protection. Mice that received the multivalent vaccine had a 100% survival rate.
100%.
Yes. And it wasn't just that they barely survived, they maintained completely stable body weights throughout the entire monitoring period.
Why is body weight the primary metric there? Why not just track survival?
In animal models, especially mice, rapid weight loss is the primary clinical sign of severe systemic infection. If they are losing weight, it means the virus is replicating and causing internal damage, even if their immune system eventually fights it off and they survive.
So they are getting sick, just not dying.
Right. But the fact that their weight remained perfectly stable means the immune response was so incredibly robust that the virus never even gained a foothold. The disease was stopped before it could start.
And the control group of mice, what happened to them?
The control mice, who received either a harmless saline solution or just an empty lipid nanoparticle with absolutely no mRNA inside, all rapidly lost weight. They developed severe hemorrhagic symptoms and succumbed to the infection within roughly a week. The contrast couldn't be starker.
Hold on, I want to talk about the timeline of this protection, because this specific detail blew my mind when I was reading the paper. They didn't just vaccinate the mice on Monday and expose them to the virus on Friday, did they?
Not at all. They wanted to rigorously test the durability of the immune memory.
How long does the shield last?
Exactly. They tracked the antibody levels in these mice over time, watching them peak about a month after the booster shot, and then gradually plateaued into a highly stable, steady state.
And then?
Then they waited. They exposed the mice to the lethal virus a staggering 17 months post-vaccination.
17 months. I actually had to look this up in mouse years. That is practically their entire natural lifespan. They essentially gave these mice lifelong immunity from a single vaccination regimen.
It is a phenomenal, almost unprecedented result for this class of virus. To put that into human public health terms, achieving durable immunity that lasts for years without needing constant yearly boosters is the absolute holy grail of vaccinology.
Especially for a disease like Ebola. I mean, we are talking about outbreaks that often happen suddenly, explosively, in incredibly remote areas with very little infrastructure.
The logistics are usually a nightmare.
Exactly. Trying to deploy a massive vaccination campaign, maintaining the ultra-cold chain required for mRNA vaccines, getting medical teams into these regions in a matter of days, it's incredibly difficult.
It is.
But if you have a multivalent vaccine that provides durable immunity for years, you could theoretically vaccinate populations in high-risk areas proactively. Having a population with pre-existing durable immunity changes the entire epidemiological math.
It completely flips the script.
It stops an outbreak before it can even become an epidemic.
Precisely. It literally breaks the chain of transmission at day 0. The study also provided some really fascinating insights into how the vaccine cleared the virus from specific organs, which tells us a lot about how the immune system is deploying its forces during an infection. They looked closely at the liver and the spleen.
Why focus on those two organs specifically?
Well, they are major targets for the Ebola virus, where it replicates heavily, and they are also massive hubs for immune activity. Now, lipid nanoparticles, by their very biochemical nature, naturally tend to accumulate in the liver after they are injected into the bloodstream.
That's just where the blood flow takes them.
Mostly, yes. They are eventually taken up by Kupffer cells, which are specialized immune macrophages that live permanently in the liver.
So the delivery vehicles naturally traffic straight to one of the primary battlegrounds.
Yes. And this natural targeting actually aided in clearing the hepatic, or liver, viral loads. In fact, the researchers noticed something highly unexpected. Even the mice that received the empty lipid nanoparticles, the ones with zero viral mRNA inside, showed a very slight temporary reduction in liver viral load.
Wait, why would an empty fat bubble fight a virus? It doesn't have any instructions in it.
It wasn't fighting the virus specifically. The lipid nanoparticles themselves cause a localized, nonspecific immune inflammation.
Like an irritation.
Exactly. The liver's immune cells sense these foreign lipids, get mildly irritated, and ramp up their baseline antiviral state just in case. It's not nearly enough to save the mouse from a lethal dose, but it shows how the delivery vehicle itself interacts with the organ's native immune environment.
That is fascinating. The vehicle itself primes the battlefield. But the spleen told a completely different story, didn't it?
It did. The spleen is a massive hub for T cells. It's essentially a military barracks for the immune system. To clear the virus from the spleen, the animals couldn't rely on that localized lipid inflammation. They relied incredibly heavily on that nucleoprotein-driven cellular response we spent so much time breaking down earlier.
And the researchers actually proved this in a really dramatic experimental way.
They did.
Yeah.
They performed a highly specific T-cell depletion experiment.
How does that work?
Right before exposing a group of fully vaccinated mice to the lethal virus, they injected them with a specific chemical antibody that temporarily depleted all of their CD8-positive killer T cells.
They essentially benched the entire internal security force.
Yes, they removed the assassins from the equation.
So these mice had the strong antibody wall from the glycoproteins, but no killer T cells to back it up. What happened?
The protection absolutely plummeted. Without those T cells, the virus was able to take hold in the lymphatic organs like the spleen. The mice lost significant weight and the mortality rate spiked. This proved conclusively that the antibody wall was not enough on its own. The nucleoprotein's ability to summon those killer T cells was absolutely essential for clearing the virus and ensuring ultimate survival.
It perfectly validates the entire premise of the four-part multivalent recipe. You absolutely need the paint job to build the initial wall, and you absolutely need the engine block to train the assassins for cleanup. If you remove either one, the virus wins.
Exactly. And to further validate the broad-spectrum claim of this paper, we have to talk about the cross-strain success.
Right, because it wasn't just Zaire.
No. The multivalent approach was not just successful against the Zaire strain in mice. They also rigorously tested it against the Sudan virus.
The exact strain that caused the 2022 Uganda outbreak.
Yes. Now, for this, they couldn't use mice because normal mice aren't easily infected by the Sudan strain. The biology just doesn't line up. So they used a completely different animal model. Golden Syrian hamsters.
Okay, so a slightly larger rodent model.
Right. They exposed these hamsters to a lethal guinea pig-adapted Sudan virus.
And the results? Did they mirror the perfect protection in the mice?
Perfectly. The multivalent vaccine group maintained perfectly stable weights, showed absolutely no clinical signs of disease, and when they tested their blood, the vaccine had eliminated all circulating virus.
Complete protection against a completely different strain, using the exact same all-in-one vaccine regimen.
It is incredibly impressive data.
It really is. It feels like a massive historic leap forward. But as always on this show, we have to stay strictly honest with the listener. We need to look critically at the study design constraints.
Right. No study is absolutely perfect.
Exactly. And if we want to truly understand the science, we need to ground our enthusiasm in the biological realities of the experiment. This brings us to the limitations of the research.
And the most notable limitation in this specific paper involves exactly how they tested the Bundibugyo virus.
Right. So we had mice for Zaire, hamsters for Sudan. What model did they use for Bundibugyo?
Well, they ran into a significant biological roadblock here. The Bundibugyo strain does not cause severe overt disease in normal, healthy mice.
Their immune systems just fight it off naturally.
Yes. Their innate immune systems handle it too well. If you inject a normal mouse with Bundibugyo, it might get a little sick, maybe lethargic, but it won't die, which makes it completely impossible to study whether a vaccine actually prevents a lethal outbreak scenario.
You can't prove a vaccine saves a life if the virus wasn't going to kill the animal in the first place. The data would be meaningless.
Exactly. Therefore, to test the vaccine's efficacy against Bundibugyo, the researchers were forced to use a highly specific, genetically modified mouse called an IFNR knockout mouse.
IFNR knockout. Let's explain what that means, because this is crucial context for understanding the limitation. IFNR stands for interferon alpha/beta receptor.
Right. Interferons are absolutely crucial signaling proteins in our bodies. They act as an early warning system, kind of like a biological fire alarm.
So when a cell gets infected, it pulls the alarm.
Yes. When a cell is infected by a virus, it releases these interferons into the surrounding tissue. This basically tells all the neighboring healthy cells, hey, there is a virus actively replicating here. Lock down your borders and heighten your antiviral defenses.
It's essentially a cellular neighborhood watch system.
Exactly. But these specific knockout mice have been genetically engineered in a lab to lack the intact receptor to actually hear that warning signal.
So the fire alarm is ringing, but their cells are completely deaf to it.
Precisely. Because they lack this receptor, their innate immune responses are severely impaired. That is the only reason the Bundibugyo virus can replicate fast enough to be lethal to them.
So why is this a limitation for the study? It seems like a clever workaround.
It is clever, but it raises a very important question regarding translatability to humans. Because these mice have a deliberately compromised immune system, they represent a surrogate model.
They aren't normal.
Right. They do not perfectly mimic a healthy, robust human immune system.
We are essentially observing the vaccine's efficacy in an artificially weakened host.
Yes. Now, to be completely fair to the researchers here, the fact that the multivalent vaccines still conferred strong protection and significantly reduced viral loads, even in these severely immune-compromised mice, is actually a massive testament to the vaccine's raw potency.
It worked even when the mouse's own immune system was fighting with one hand tied behind its back.
Exactly. But from a strict, rigorous scientific standpoint, it remains a limitation. It is an artificial scenario that we have to acknowledge.
And there's another caveat explicitly mentioned in the paper's discussion section that I found really fascinating, and it goes all the way back to our all-in-one lipid nanoparticle design, the antigen overload caveat.
Yes, this is a very real biochemical construct of the platform. We are cramming 4 distinct mRNA blueprints into a single lipid nanoparticle. When that microscopic fat bubble merges with a host cell, it dumps all 4 sets of instructions into the cytoplasm at the exact same time.
And the cell's ribosomes, the microscopic factories that actually read the mRNA and physically build the proteins, suddenly have to process all of this simultaneously.
Exactly. There is a biological upper limit to how much exogenous or foreign protein a single cell can produce at one time.
The factory only has so many workers.
Right. If you flood the cell with too many blueprints, the machinery gets completely overwhelmed. This can lead to severe cellular stress. It can trigger what's called an unfolded protein response, or it can simply result in suboptimal protein expression, where the cell does a mediocre job building all 4 proteins rather than an excellent job building just one.
So the researchers really have to find the perfect Goldilocks zone. Enough mRNA to trigger a robust immune response, but not so much that you crash the host cell's manufacturing plant.
Precisely. And the authors noted that optimizing the exact molecular ratios of these 4 mRNAs inside the nanoparticle is an ongoing complex challenge. They found a ratio that worked beautifully in mice and hamsters, but that specific ratio might need to be carefully adjusted for larger mammals.
Which logically leads to the necessary next steps for this research. What happens now? I mean, we have incredible data in small animals. Does this go straight into human arms for phase 1 trials?
Absolutely not. Before this multivalent platform can even be considered for human clinical trials, it must be rigorously validated in higher-order animal models, specifically testing in non-human primates.
Why are non-human primates the mandatory stepping stone here? Why not just jump from hamsters to humans?
Because their immune systems, their specific cellular receptors, and the entire disease pathology of how filoviruses actually attack their bodies closely mirror human infection.
They're biologically closer to us.
Right. A mouse immune system is a great sandbox for proving a concept, but a non-human primate is the true biological proxy. What works in a mouse or a hamster is a crucial, necessary proof of concept, but non-human primate data is the absolute required bridge to ensure it will be both safe and effective in human trials.
So let's pull back from the specific data for a moment and look at the broader expert angle. How does this specific paper, with all its elegant molecular engineering, the MOLDEL deletions, the synchronous LNP delivery, and this robust animal data, how does this fit into the broader trajectory of virology and our global outbreak preparedness?
If we connect the highly specific findings of this paper to the bigger picture, it validates a massive scientific consensus that is fundamentally shifting the entire field right now.
Bigger than just Ebola.
Much bigger. This study does not just offer a potential multi-strain Ebola vaccine. It acts as a profound, undeniable proof of concept for the rapid adaptability of the multivalent mRNA platform against the entire filovirus family.
The entire family.
Right. We are already seeing parallel breakthroughs using this exact same mRNA logic for the Marburg virus, which is a close, equally deadly relative to Ebola.
So the overarching perspective here is that mRNA antigen engineering, is finally allowing us to transition from that reactive posture we talked about at the beginning.
Exactly. No more scrambling to sequence a single strain and build a bespoke vaccine while an outbreak is actively spreading and killing people.
We can finally move to a truly proactive posture.
We can rationally design broad-spectrum defenses that anticipate multiple viral variants simultaneously. We are literally laying down the architectural foundation for a true panfilovirus defense strategy.
So what does this all mean for you, the listener? Let's blend it all together into a closing arc.
The main takeaway is synthesis.
Right. By engineering a single lipid nanoparticle to synchronously deliver both the rapidly mutating outer spikes and the highly stable internal core of 3 distinct Ebola strains, scientists have created a beautifully balanced, broad-spectrum vaccine that provides complete, long-lasting protection in animal models.
The actionable insight here is a mental model that you can apply to all future health and science news.
How to read the headlines.
Exactly. Whenever you read a headline about a new viral outbreak or a new vaccine entering clinical trials, ask yourself, is the medical response targeting a single highly mutable surface protein? Or are the researchers actually combining targets to create a multi-layered cellular defense?
Are they just painting over the license plate, or are they also going after the engine block?
Exactly. Understanding that core biological distinction separates a superficial, surface-level reading of the news from a true, deep understanding of immunological strategy.
We will be continuing our exploration of lipid nanoparticles in our next deep dive. We are actually going to pivot from vaccines and explore how these exact same microscopic delivery vehicles might soon be used to deliver CRISPR gene editing tools directly into the human liver to cure inherited genetic diseases.
It is a wild, rapidly evolving frontier in medicine.
But before we go, consider this final provocative thought. If this multivalent mRNA platform can seamlessly process 4 distinct antigens to provide a robust shield against an entire family of deadly filoviruses, what is the ultimate biological limit of the lipid nanoparticle?
How far can we push the platform?
Right. Could we eventually design a single universal panvirus injection? Imagine a single shot that forces your cells to express the conserved core proteins of dozens of completely unrelated pathogens, from coronaviruses to influenzas to Ebola viruses, equipping your immune system with a comprehensive threat library before a spillover event even happens.
It completely redefines what we consider possible in the future of preventive medicine.
It really does. Thank you for joining us on this deep dive into the vanguard of vaccine technology. Stay curious, and we will see you next time.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.